Hello everyone, welcome to our webinar. We will start our webinar in 1 minute as people are still dialing in. So, hello everyone. We will start our webinar in a few seconds as I could see that people are still dialing in. So, so, so hello everyone and welcome to our today's webinar. My name is Martina Witzenbacher, and I'm a portfolio manager for chromatography consumables at Avantor. Together with my colleague Didier Semper from marketing, we will host today our July chromatography webinar with the following topic: Keep pace with advances in therapeutic protein characterization. Our today's speaker will be Dr. Ken Cook from Thermoscientific. Dr. Ken Cook's industry experience spans over 30 years with numerous publications in diverse fields including environmental, oil, food safety, and adulteration, proteomics, protein chemistry, and metabolomics and lipidomics. Currently, his works involves the support of biopharmaceutical applications to characterize proteins and oligonucleotide-based biotherapeutics. This involves ongoing collaborations with biopharmaceutical industry and academia. So it's a great pleasure for us to welcome him to our webinar. Before I hand over to our today's speaker, I would like to give you a couple of housekeepings information from my side. So if you have any questions, and I'm sure you will have many questions during the presentation, you can enter them anytime by typing them into the question tab, which are on the right side of the menu screen. We will have some times after the session to ask some questions directly to the speaker. For questions which requires further work or further research, we will come back to you after the webinar. If you have some additional questions after the webinar, please do contact us at webinar@avandor.com. You will get this email address in a mail afterwards. So during this webinar, Dr. Ken Cook will explain developments and tips for new workflows for intact protein mappings, characterization of proteins, new column chemistry for charge variance analysis with pH and salt gradient, and a lot of other topics. So far, so good. We will now start the presentation. So Ken, the stage is yours now.
Yeah. Can you hear me all correctly?
Yes, I could hear
Yeah,
you.
that's fine. Thanks for inviting me to give this talk. And in this talk, I'd like to go through some new workflows for intact and peptide mapping characterization of proteins. A lot of this work is supported by some new column technology that's just come through. So you'll see some new column technologies for ion exchange, for size exclusion chromatography, and a new one for peptide mapping. We'll also look at some of the sample preps for where we've got smart digest for protein digestion. And then we'll look at questions afterwards. Now, Initially, what we're really looking for here is, and the talk's going to center around monoclonal antibody characterization. And from the monoclonal antibodies, they're a therapeutic. They've got to be analyzed so they know they're safe and they actually work. So the special quality or critical quality attributes we look at, the most common ones that people look at are deamidation, oxidation, because they're an indicator of the monoclonal antibody degrading. But we've also got to look at glycosylation and making sure the pattern and the way that monoclonal antibody is made in a controlled fashion from batch to batch. You do get point mutations and other impurities, disulfide bonds, just to make sure the protein is folded in the correct manner. Now, when we look at total biopharma analytical workflows, the ones in the green here on the top left, we're going to talk about them in this seminar. So we're going to look at intact protein analysis. We can do that by charge variant analysis. We can do it by size exclusion for the aggregates. And then we're going to look at the glycan analysis as well, but we're going to look at the glycan analysis through either the intact protein analysis or peptide mapping. We can do glycan analysis by released and then released and labeled, and that's usually done by helic chromatography on an AccuCore amide helic column. We're building up a lot more therapeutic analysis methods, but again, some of these columns are mainly the same ones I'm going to talk about now for proteins. There's a big interest in oligonucleotide therapeutics. You've got the bigger messenger RNA therapeutics and also the shorter ESO antisense therapeutics. Now, there's a good range of columns for that, and the DNA pack RP, which is the reverse phase ion pairing column, but you've also got the DNA pack PA200s, which are the ion exchange versions. And during the seminar, we'll be talking about new column for peptide mapping and how that's used in the multi-attribute method, and it can also be used, same column, for whole cell protein analysis. Now, when we look at protein characterization, particularly of monoclonal antibodies, there's two main routes. The first route is to go intact analysis. That requires no sample preparation. The monoclonal antibody is going to be injected directly into the column. And in this case, we'll be using columns such as the MABPAC RP column, and that will give you the denatured intact profile. And then if you want the native intact profile, and we'll talk about the differences of that in a later slide, there you've got the size exclusion columns in the MABPAC SEC. The other common one would be the MABPAC SCX. Now, both of these columns have new columns in the ranges. We've got a new ProPack column, which we'll discuss later on for charge variant analysis. And we've also got a shorter, faster size exclusion column. Now, if you look at peptide mapping, there's a new peptide mapping column come out called the hypersigal peptide HPLC column. We'll tell you about that one. But before you get to the column, you've got to do a digest. So I'm going to show you some new techniques for that digest that makes a digest much easier to do. But after you've done that, whether it's a protein level or the peptide level, the LCMS platform is the same. If you're using a MABPAC RP column, it's actually the same eluent you'd use for peptides. As soon as you go to size exclusion and ion exchange, the elements are going to change. But the profile for the LCMS, that platform is going to remain the same. And then, of course, you'll do the number crunching and the qualitative analysis in the software packages. Now, to delve a bit deeper into the multi-attribute method, this started life and was first published in 2015 by Amgen. Now, so that's quite a while ago now. But what it was designed to do was to take away a lot of the conventional HPLC methods, and there's a multitude of HPLC methods that are used to traditionally analyze a monoclonal antibody. You look at size exclusion, charge variance, hydrophobic interaction chromatography, helic, peptide mapping. There's a lot of individual methods. So instead of doing all these individual methods, try to get one method and incorporate multiple attribute quantitation into that single method. And that was done at the peptide level. Now, because it's at the peptide level, you're doing a digest. So you're basically smashing the protein to bits. So you're not going to get any information about aggregation, and you won't get any information of fragmentation because you've already smashed the protein to bits. But you will be able to see the deamidation and the oxidation because you'll be able to pick up those specific modified peptides in the digest. Succinimide's the same, lysine truncation, a whole list of things that would normally take lots of methods, you're doing in one run. And the ones at the bottom I want to point out is disulfide mapping. Yes, you can do that, but you change the digest so that you don't use either TSEP or DTT to break the disulfide bonds. Those disulfides have to remain intact, and then you'll get the site which peptides are bonded to each other. And again, with all these techniques, because you're at the peptide level, it gives you the site of modification. Now, we'll give you an idea why I've just gone through that, because now I'm going to use the same type of slide, but instead of the peptide level, we're now going to look at the protein level. Now, at the protein level, well, we're not doing a digest, so you can get the aggregation and you can get the fragments. So here it's very, very complementary to the peptide multi-attribute method. You're getting the things out now that you can't get in that method, but you'll still be able to confirm deamidation, oxidation, lysine truncation, the glycosylation sites. They'll all still be there for you to look at the intact level. But you won't get the site of the modification because it's at the intact level. You'll say, yes, that protein's got an oxidation site, but you won't know where the peptide mapping would. And then disulfide mapping, of course, you're not going to get them either because you haven't denatured the protein. But the two methods of doing multi-attribute method technology can be done at the protein and the peptide level, and they're complementary. We're going to go into first looking at the peptide level, and what do we mean in the concept of MAM? Now, what you've got here is a therapeutic protein. First off, we're going to go and do a digest, and I'll go through the digest technique later on that we're using here. Once you've done that digest and you've got lots of peptides, you'll put on a C18 column to do LCMS analysis, and then you're going to get a nice profile or a peptide profile through the chromatography here. Now, that's what you'd regard as stage one. Now, because you've got that profile there, and you'd usually do MSMS on that profile as well. So each of those peptides will be characterized. They'll be characterized, sequenced, and the parent mass of that peptide will be known as well. So whether it's got a modification, and you look at the modified and the unmodified peptide. Now, if you want, once you've characterized that at the start, you can actually put that into a targeted list and find those peptides and quantify those peptides just at the MS level. And that will give you a nice quantitation, and that's what the MAM method's all about. You can do an untargeted approach again and just characterize each peptide, and there is something called new peak detection where you've got a nice profile pattern up the top here. What happens if you've got an extra peak? That extra peak must be quantified, it must be identified, and find out what's caused that, because it shouldn't be there. It's now a different impurity you didn't know, and that's determined as new peak detection. Now, what's the challenges in doing this with peptide mapping? Now, first of all, you've got all these things you're looking at. The quality attributes you're looking for, we want to find those oxidized peptides. We want to find the deamidation peptides, but we also need the unmodified version so you can tell how much deamidation's gone on. And again, with all the impurities and disulfide mapping, your peptide map's got to make these attributes available. So really, when you're looking at the peptide map, you've got a variety of post-translational modifications that you're looking at. Some of them are at very, very low level. You haven't got a problem with the amount of sample you can load onto the column, but some of these peptides that are modified are in low abundance compared to the high abundant peptides that are unmodified. So you've got that ratio that you've got to be able to determine through your column technology. It's also, you need a high resolution separation. Because of that, some of these modifications are hard. Oxidized peptides tend to move quite a distance from the unmodified version. Deamidation, that's a different ball game. It's not easy to separate a deamidated peptide from a non-deamidated peptide by reverse phase. You need a good column for that. You need some good high resolution separations. And of course, it's got to be compatible with mass spectrometry. So you don't want a column that's bleeding things off the column that's just going to cause trouble and noise. It's got to be a nice clean column for mass spec, and you need that column to be reproducible. You've got to use that column again and again and again for years. And when you buy a new column, it's got to be the same, because this method's now in an SOP. And you want to look at poor carryover, and I'll show you some instances of carryover in the later slides. Now, first of all, we're going to look at the digest. Now, the digest, traditionally, you'll do an in-solution digest. The protein's nicely folded, and an enzyme such as trypsin is not going to cut that protein up unless you unfold it. So you've got to put a denaturant in. That'll be your urea, or it'll be guanidine hydrochloride. And then you really want to break the disulfide bonds up. So now you're doing a disulfide bond reduction with DTT or TSEP. And then, because you've now got to remove that denaturant, because if you add trypsin at that stage, the trypsin is going to denature as well. So you've got to remove that denaturant, but you don't want the peptide to fold back together, so you're going to reduce and alkylate the peptide bonds that you've just broken, so they can't come back together. Then you'll do the digest, but that's a lot of steps. Now, in this instance, what we've done here, we've actually put trypsin on a magnetic bead. We've got them on normal beads, but magnetic beads are becoming the one that's most commonly used, because you can automate at that stage. So you've got a lot of trypsin on the bead. Now, the bead itself is it's porous. So you've got trypsin inside the pores. It's high capacity. You're using now about 1,000 times more trypsin than you'd use in an in-solution digest. Now, if you do an in-solution digest, you need to know the protein concentration. Then you'll add the trypsin amount to, say, 25, 1 in 25 ratio or 1 in 50 ratio. But if that ratio changes, your digest changes. So you have to do a protein concentration. Now we've got the trypsin in excess. Because it's in excess, we don't care about that protein concentration, because the trypsin's in excess anyway. And if you add too much of the beads, doesn't matter, because it's in excess. If you add too little, that doesn't matter either, because it's still in excess. So it makes the analysis much, much simpler to do. And if you look at what you get in the kit, all you get in the kit is a buffer that you're going to use and the beads. It's only two things you need. The only additional thing will be TSEP, and that's usually, we use bond breaker, and that's just in solution. You don't have to make it up. So that's, again, it's an easy thing to do. So really what you're looking at here, you've tried to get the advantage of, you've simplified. It's much, much less steps. There's fewer reagents there, and you've increased the robustness, because this thing's very, very reproducible. You'll get 5 different chemists to run it. Some of them untrained, they'll all still get the same result. Whereas an in-solution digest, it's much more difficult to perform. If you look at it in more detail, the trypsin you've got on these beads is heat stable. So we do the digest at 70 degrees C, and when you're above 70 degrees C, monoclonal antibodies will denature. So you've now denatured the monoclonal antibody, but you've denatured it in conditions where the trypsin's still stable. So you're doing the digestion under the denaturing conditions, which is impossible with an in-solution digest. So that's why, one of the reasons it's simple. And we can also do the reduction at the same time. Now, that can cause problems, which we'll look at now, because that reduction will start to break the trypsin up. And even though the trypsin's heat stable, under those reductive conditions, the trypsin's only going to last about 15 minutes. Now, because it's on a magnetic bead, you can now automate that. That's an example of a Kingfisher. It's about the size of an inkjet printer, but it allows you to automate and move those magnetic beads around. Now, what's the problem with the trypsin and the digest? Well, if you do a single step digest, as I've just described, your trypsin will start to denature after about 15 minutes. And because of that, you'll get miscleavages. Now, the miscleavages here are represented in these red dots, and you can see there's a lot of red dots there compared with the one above, which is the new 2-step digest. You've removed a lot of those miscleavages. If you compare that with a conventional in-solution digest, you still get miscleavages with the conventional digests as well. But now when you look at it in more detail, this 2-step digest is actually, it's hardly giving you any miscleavages. You can always expect miscleavages with trypsin. There's some sites trypsin does not like to cleave at, and that will always be the case. So you've got a little bit there, but when you look at the pink and the orange ones, these are multiple miscleavages. You're only really seeing at the most one miscleavage, But when you look at the one-step digest, you've got quite a few of these. And even in the conventional digest, there's more miscleavages. Now, why do you not want miscleavages? Because basically, if you've got two miscleaved peptides, you've now got a quantitation problem. You've got to build them two together. If you've got a perfect digest and only one peptide, it's much, much easier to quantify. Now, the work I'm showing you was done by Dan Bach Christensen's group in Symphogen in Copenhagen. Now, what happens with the two-step digest, we know that trypsin's going to die a little bit after 15 minutes. So we take the spent digest out, then your monoclonal antibody is already in bits, so we don't need to be at 70 degrees C anymore. We drop the temperature to 40 degrees C and complete the digest by adding fresh trypsin. And that's what's giving you the good results. Now, those results come with a problem, which we're hopefully going to resolve with a column later on. If you look at the one at the left, which is the double digest, the two-step digest, you've now got enormous cleavages, and you've got this tiny little peptide, which we're depicting here in the red circle. That's very hydrophilic, and it's going to go straight through most C18 columns. It's not going to bind, and you're going to miss it. So you're no longer going to get your 100% cleavage, sequence coverage down at 98. And that can happen with a few different sites. It's not just one site. So you could drop your cleavages. Whereas if you've got miscleavages, that small peptide there is picked up in the miscleavage peptides. So that's why you're up at 100%. So basically, the more miscleavages you've got, the more coverage you've got, but you've got a quantitation problem. So you'd like to get both. This is where the new peptide column comes in. This peptide column is a Hypersil Gold C18 column. Now, we produce that silica. It's very carefully controlled. Everything that happens to that column is under control. We're not buying any OEM silica in and functionalizing it. Everything's done on site. So we're much, much in control of the column. And this column now, instead of just being tested with small molecules, it will come tested with a peptide separation, where we do look at critical peptide resolution, so we know the resolution's there, and we'll also look at hydrophilic peptide retention for the very reasons I've just spoken about. Now, is it better than any other column? And a lot of people will say, well, it's C18 column, they're all more or less the same. All C18 columns are not the same. The silica will be different manufacture. That'll have different attributes on the silica itself, different ion contamination, cyanol groups. Silica is not the same. Now, when you look at all the C18 columns, there's a competitor column we've tested against at the top. And again, that looks quite nice. It's giving you good reproducible peaks, and the peaks are relatively good shape. But we'll go into the detail a bit more on competitor 1 column. Competitor 2 column, yes, it looks okay, but it's got noticeable retention time shifts. That column's just not stable enough. Now, quite often, polar embedded columns are pushed for peptide resolution because you can run them at 0% acetonitrile. And it doesn't matter whether you run them at 0% solvent, they do not bind hydrophilic peptides. You can see the profile comes out earlier than the other columns, and you lose a lot of hydrophobic peptides at the front. These polar embedded columns are not good for peptide mapping. The one at the bottom is our new Hypersil Gold column. It gives you that hydrophilic retention, and every single one of those peptides comes out on top of each other. Now, when you look at the peak shape, the competitor 1, which looks good from the profile, once you start looking at some of the peaks, yes, you've got some of them are nice and sharp, but some of them have got tail-in problems. And that tail-in problem prevents deamidation quantification in quite a few sites. So again, you can't use that column easily because you'll lose some of the deamidation critical attribute peptides that you're looking for. And again, competitor 2 looks quite clean, but when you look at the Hypersil Gold peptide, you've got really nice sharp peptides. And if you look at the counts, because the peak sharp's sharper, you actually get more sensitivity, and the sharper the peak, the easier they are to quantify. Now we're going to look at here deamidation variance. That's only 1 Dalton difference, so it's much better if you can actually separate that deamidation variant from the non-modified. In peak 1 here is the unmodified, but peak 2 and 3, you can see you've got a very good separation here for deamidation, and that's selectivity. Not all C18 columns will give you that selectivity. And there's a bunch of deamidated peptides that we're separating and quantifying very, very well with this column. Now, here's an example where we've taken some rituximab and nismab peptides. Now we're looking at lot-to-lot reproducibility. This column's got to be the same each time. And as I said before, we can control the silica production, the packing, everything's controlled with this column. So the lot-to-lot reproducibility is very, very good. And that's an example here on the top table where you look at the peak asymmetry, the same all the way through. You've got good peak asymmetry. The retention time shifts are ridiculously good. They're bang on top of each other. And the area counts again the same. But when you look at column to column variability, you've got lot 1, 2, and 3 here, and they're all more or less the retention time stability, they're bang on top of each other. This number here from lot to lot variability in RSDs, people would be happy if they could get that on one column. This is 3 columns. So that's how good they are. And then if you're looking at modifications, does it pick up the modifications? Well, here we're looking at some glycosylation variants. And again, when you look from lot 1, 2, 3, 4 here, the glycosylation data you get from them exactly the same. Again, another one in the middle and another one at that side. But basically, it doesn't matter which column you use. It's going to be, whether when you finish the first column and you need a new one's going to be exactly the same. So if you look at the specifications we've got here, it's got a good pore size for these larger peptides. It's 174 angstroms. It's a small particle size to give you that really sharp, good peak shape. And it's tested now for peptides. When you get this column, you know it's been tested and it's been passed some very severe test criteria before it comes into your hands. Now, once you've done the column, you've got to look at the sample handling solutions. And what we tend to use now is these, we're using a lot of polypropylene vials, but it's not just any polypropylene vial. These are low bind vials. Some of the peptides you're going to get can be very hydrophobic, and you do a nice digest, your solution goes into the vial, but your hydrophobic peptides don't come back out. So these vials are special vials to make sure that you retain and pull out those hydrophobic peptides. The plates are the same, whether you're using the short of the plates or the deep well plates. And in addition to the plates, there's a nice web seal on the top, because it's easy to put a nice short stop cap on a vial. It's not so easy to actually cover a vial at 96 well plate. And these ones cover them really well, and they work just more or less the same as a seal in a standard vial. So peptide mapping, and here in the title I'm saying, have we been doing it wrong for many years? If you're going to do peptide mapping and use C18 columns, why do we use C18 columns? Because everybody uses C18 columns, so we'll just use the same as everybody else. Now, we had a problem. Again, this was Dan Bach Christensen's group in Symphogen. Now, there was a problem with a very, very hydrophobic peptide. Some hydrophobic peptides did give some tailing problems anyway, but this particular peptide was giving you a really bad tailing. This is an extracted ion chromatograph for the peptide itself, and you can see it's tailing all the way through the length of the column, or the length of the run after the peak comes out, and it's a horrible peak shape. And then when you do a wash, you get a load of carryover coming from the wash, and then a load of wash with the descending wash. Now, this is a symptom of the fact that all resins will swell with and shrink with acetonitrile concentration, and that's why you see them on the front of the wash and the back of the wash. So people doing really long washes of solvent for 10 minutes at 100% solvent, nothing's coming off there. It comes off on the downside and the upside. You're much better off doing sawtooth gradients. This is an example of the C18 column, but now if you move to C4, so this is the hypersil C4 column, you've got a lovely peak shape for that one, virtually no carryover, and even that really nasty hydrophobic ones, now give them a peak we can now quantify. There's a tiny bit of carryover, but the actual wash gets rid of it. Now, this is a hydrophobic peptide, and we're using a C4, so you can think, well, the C4 is not going to bind the hydrophilic peptides. It actually does. Now, that's an example of the C4. That's an example of the C18 run. They're very, very much the same, and is binding the hydrophilic peptides, because the C4's actually got a higher carbon load than C18. You can get more C4 chains on a resin than you can a C18 chain. So we're not losing the hydrophilics. So basically, you're looking at this and thinking, why are we using C18 columns for peptide mapping? And I know in Symphogen, they don't use C18 columns anymore. They go straight to C4. So that's a possible and something to think about if you've got a problem with hydrophobic peptides. But again, when you look at selectivity, the selectivity is very, very good. And you've got to remember, if you're doing that digest, you need to be able to retain those hydrophilics. I'm going to go now to look at native work. Now, if you put it on a reverse phase column, and again, the column we look at here is the MabPack RP. The MabPack RP is a polystyrene column with divinyl crosslinks. It's got a very wide pore size for bigger proteins. Now, the bigger the protein you get, the worse the carryover. Some people use C4. Will you get a peak for C4 on protein? Yes, you will. But if you run a blank afterwards, you'll still get a peak, and then you'll probably still get peaks for the next 3 or 4 blanks. The carryover for big proteins on C4 silica resins is enormous. When you use the MabPack RP, the carryover is virtually zero. So the polymer resins don't give you that carryover that a silica resin would on a big protein. But if you use that, the MabPack RP, you've denatured your protein, you're insolvent, and you'll get a big charge state distribution around about 3000. Now, if you pick any one of those charge states, you'll actually see the glycan distribution underneath the charge state. Now, if you go to native mode, and here I'm talking about ion exchange or size exclusion, you've now got the protein folded, so the only charge is accessible on the outside of the resin, outside of the protein, rather than every charge being available on a denatured protein, so you get less charges. So your mass to charge ratio goes up, but your spatial resolution goes up with it. The distances between your charge states are much more resolution than you've got here on a denatured one. And we're looking at 6,000 instead of 3,000 here now for the charge states. Now, when's that important? Well, here's a simple one. You can see there's a big gap in the middle there, but that's giving you the same glycan profile here on one charge as you would on denatured. But what happens if you suddenly got more glycosylation? If you've got sialic acids in there and it's more heterogeneous, these are now going to spread across and then interfere with the next charge state. If you start putting a drug on, such as an ADC, that again is going to give you more charges. And again, these are going to overlap each other. So the more spatial resolution you've got, especially with complex proteins, the better you are off. So native's good. Now, this is an example of doing a multi-attribute just with the size exclusion column. This is a case where we've run the size exclusion column, that's your monomer, and you're looking at your aggregates here. So you can get your aggregate quantitation by UV. And when you look at the MS, again, take one charge state, so now you've got the glycan distribution. And you can see that's a relatively complex glycan distribution, and it's beautiful. So this is using size exclusion. And here we can look at the quantitation ions. So you can quantitate these ions from this charge state, and now we can actually see all the different glycoforms quantitated. So you'll get the intact mass, the glycan profile, and the aggregate in one run. Now, if you want to do that faster and high resolution, this is where the new column comes out. This is the native pack OBE SEC column, and it's really there as a fast desalter. Now, this column here, it's you're looking at the desalt happening in a very short space of time. It's 3 minutes from run to run. But here you haven't got as much separation. So this is your protein coming out first. You're going to divert to waste after that protein's out and take that rubbish out of the mass spec. So this is your non-volatile salts here. This is the intact protein that's been desalted in front, but it's a quick run. And again, you'll get the glycan distribution. It's going to just happen a lot faster. This is an example of doing multiple NIST-MAB analysis, and again, one after the other, just shooting straight through, simple setup, and after you've gone through the column, you've just got a short divert valve before you go to the mass spectrometer. Salts go to waste in the mass spec, but you'll get some beautiful traces from this, and you'll get your intact mass and your glycan profile quickly. Now what I'm going to go into is a brand new column. Now this is a column that it's monodisperse. All columns are usually polydisperse. So when you see this structure on the right hand side here, you can see, yes, it's a 4 micron resin or a 3 or a 2, but they'll not all be 4. They'll not all be 2. It's polydisperse, and because of that, it packs strangely in the column. So column to column reproducibility is harder to get here because it's going to pack differently in every single column. The columns we're bringing out now, the resins are monodispersed and they just line up like little soldiers. Every single column you pack is going to be more or less exactly the same. But because you've got a monodispersed structure, the path through those beads is very uniform. The path through these beads, it's not uniform. So what happens now is you've got a uniform path, and you've got a uniform path here on the bottom, and you get a much sharper peak than if the path's more diverse because it's a polydispersed resin. That's going to broaden your peaks, and that's called the A term. So we've got better resolution with these monodispersed columns, and these comes in quite small pore sizes, quite small particle sizes. That's a limit, and we've now reached it with particle size. This is the next step where we're now going to monodisperse. Now, all the other columns you normally use are still going to be available. This is just the new format to give you that extra resolution. Now, we'll take those monodispersed beads, put a hydrophilic coating on the outside, and then functionalize it either with a strong cation exchange group or a strong anion exchange group. So you can do anion or cation exchange. The lot reproducibility on this, because they're monodispersed and the pack well, is immensely good. It's much better. But you've also got higher resolution. We'll show you some of those resolution examples later on. And again, it's robust. The operating range between the SCX and the cation exchanges, it's good. Now, this is an example of the anion exchange. Now, all these particles are now 3 micron. And this is an example of that 3 micron monodisperse, and it's just a 10 centimeter long column. Now, the column that's, even now, it's a gold standard column for the resolution you can get on charge variant analysis. That's 25 centimeters long. Now, if you look at the resolution of that one against the resolution of the shorter column, it's much better with that monodispersed resin. So you've now got much better resolution. And you can use that with a salt gradient, or you can use that with a pH gradient. I much prefer the pH gradients because they're easier to set up and you usually get better resolution. The other thing with these columns, because they're monodispersed, the re-equilibration time is much, much shorter. And this is an example here from a publication that was done with NIBIT in Dublin. And this is an example of albumin. But of albumin here, the most variance that was ever found was 50. There was 150 isoforms because you're now going direct into MS with an volatile gradient system. Now, when we're looking at this anion chromatography, there's a lot of anionic proteins coming out for therapeutic purposes now. A lot of the fusion proteins are highly glycosylated, and if they've got sialic acid groups out, they're usually anionic. And here you can see we're separating all the different sialic acid groups out from of albumin. Here you can see the phosphorylation. That's 1 phosphate, that's 2 phosphates, that's no phosphate. So you've now got a separation on charge. And again, you'll get the charge profile. You've got a lot of resolution here because we're in the native form. And even though you've got a heterogeneous glycan pattern, you've still got a lot of resolution between the charge states. This is an example now of a fusion protein. And again, that fusion protein, it's very heterogeneous in terms of the glycans. You get lots of different peaks separated, but every single one of those peaks gives you a beautiful charge variant profile. It gives you a lot of a beautiful MZ profile in a charge distribution. So that's easily deconvoluted. If that came out all in one lump, so if all these peaks were together, you'd get a mess that you couldn't deconvolute properly. But because you've separated those anionic variants out, and each of those variants are coming at the mass spec one at a time, you've now got the ability to actually analyze these difficult to analyze fusion proteins in a way you never had before. This is an example now of the more common way of doing it with a monoclonal antibody. Here we're using cation exchange, but again, the 3R resin. And you can see that's a beautiful separation. And again, this is using the volatile eluent systems. Here we're just using ammonium bicarbonate, acetate, and we're using ammonium hydroxide in B. So really, it's a simple eluent, it's volatile, it'll go direct into the MS, and now you've got a really beautiful separation of the lysine truncation variants here. But you think, well, they're pretty easy to do anyway. But if you look at the tiny peaks at the bottom here, this is the blow up. And now, yes, I can do the great big ones and I can get the lysine truncation, anybody can. can I get peaks like this? A5, that's a tiny little peak we've now resolved in that column. But again, because it's coming out in a separate space into the mass spec, we can now find out what is that small peak. And that's it at the bottom. Even though it's at that tiny amount, you've still got a good mass spec trace that you can deconvolute and find out what it is. And that's what we're seeing here. A1, which is one of the big peaks, that's easy to do. So you'll get the glycan distribution from that anyway. But this is A5. That deconvolutes and the data is really nice. So where you thought we've got a tiny peak on that charge variant analysis, what is it? We can now analyze that directly into the mass spec with these columns. And to give you an idea what we're looking at here, this is adalimumab, but we're looking here, that's a double deamidation. And that's virtually invisible, yet we can still see it and identify it and quantitate it. That's a single deamidation there. You've got a sialic acid coming out there as well. But all of these profiles where you've got succinimide, they're there. And all we're doing here is looking at the highest glycoform, the most abundant glycoform, and we're actually doing single ion monitoring and actually picking these out, extracted ion chromatographs, and looking at these ones specifically. And you can say, can you tell the difference with 1 Dalton on 150,000 Dalton protein? Yes, we can, because they're coming out separately from the main peaks. You've separated them. But even if you didn't, if it wasn't perfect, you could still run it because we know where that comes out. So you can use retention time for identification as well. So yes, this can be done. And now we'll look at the summary of the 3R columns. It's much higher resolution than you'd get normally with a traditional polydispersed resin. And most of the resins coming out from Thermo Fisher will now be in this format. And there'll be more to come out, so keep an eye out for these ones. And if you're looking at LCMS, ion exchange can be a powerful tool going directly into LCMS with these volatile eluent systems. There's a few volatile eluent systems out there on the market and publications. The one I've just shown you, that works well. It's not using any salt, and the quantitation on that, because the salt concentration is the same all the way through, which is virtually none. You're around about 30 millimolar. It's very, very sensitive. That's where we're getting the sensitivity from. It's a remarkable sensitivity for this because of that. And the Orbitrap that we're using here is obviously a sensitive machine, but it allows the confident analysis of low-level sample impurities that you wouldn't have thought was possible before. And you can do it with the anion exchange and the cation exchange. Most people have concentrated on cation exchange because of monoclonal antibodies, but now you're seeing a lot more fusion proteins coming out which are anionic, and as soon as you start getting heterogeneous glycans and silation on there, it's the anion exchange that's going to help. And if you get it out in one lump on a size exclusion column, you'd never deconvolute the mess you've got underneath that peak. But once you start separating these out into different variants, now you've got something you can actually use. Now, what I'd like to show you here now is the summary of what we've got and the resources you've got. Now, the native pack OBE here, that's a fast salt desalting column it's becoming quite popular. So OBE stands for Online Buffer Exchange. The ProPack 3R columns, it's the first of a range of columns coming out. These are the ion exchange versions, and they're available in a strong cation exchange and a strong anion exchange. These are the more useful columns if you're using volatile eluents with mass spec. If you go to high and low pHs with a weak exchanger, you can lose the charge. The difference between a weak and a strong is that you can run them in extremes of pH and the charge will still remain on the column. So that's why we've gone for these strong exchangers rather than weak exchangers. They're monodisperse, and you've seen the advantages you're getting from that. The MAPAC RP columns, it's a reverse phase polymeric columns for proteins, and it's got very, very low carryover, and you've got phenomenal resolution from these things. Now, the Hypercell Gold peptide column's new, and that's been designed specifically for peptides, and most people won't put much thought into the column that they're using for peptide mapping, and I've just shown you in some of these slides that you really need to be looking at that. I've shown you a different way of using the Smart Digest, and again, the Sure Start vials, because there's little point in doing the analysis if your hydrophobic analytes are sitting in the vials. Now, These are the ordering information for all the columns. Now, I'm going to switch to questions now, but I'm going to leave this slide here open so you've got the part numbers on the screen, and I'll pass over now for the questions.
So thanks a lot. Thank you very much, Ken, for this really impressive presentation. I could really say I learned a lot today. So let me check if there are some questions in the chat. Um, I received some questions. So the first one are coming from Spain. Why have C4 columns never been used for peptide mapping before now?
That's a very, very good question. And we did it because we had a problem. But basically, this isn't just evolving around peptide mapping. People tend to copy what's been done before. And what's been done before isn't necessarily the best way of doing it. And everyone's always used C18, so people just use C18. It's the go-to column everyone will always go to. They wouldn't even think of changing it because it's always been used for this purpose. The only reason we looked at that C4 is because we had a specific problem. And then I thought, well, we'll use the hydrophilics, but you don't. And then you realize afterwards, of course you're not going to do that because you've got a high carbon load on these columns. So basically the reason C4's never been thought of before is that people tend to copy what's been done before. And now we really need to think other analytical techniques we should be thinking through a bit further as well. And I've seen a few of those coming up as well.
Okay, thanks a lot. The next question are coming from Switzerland. The question is, can you really detect the difference between a deaminated and non-deaminated protein with a mass difference of only 1 Dalton?
Yeah, no, it's always better if you can separate the peptides out. That's why we're looking at that new peptide column. And we didn't just look at it for, yes, it'll retain some hydrophilic peptides, and yes, it'll give you a peptide map. Any C18 column will give you a peptide map. The one we've got there, it separates the amidation variants, and it's the selectivity difference that does that. It's got a good selectivity. But there will always be some deaminated peptides you can't separate at all. And in that case, you've got to go up to around about 120,000 resolution, and you will see the difference. But if you're down at sort of 15, 30, you won't be able to pick out the deamidated peptide from the non-deamidated peptide unless you've got a separation. But certainly resolution will do it, but you have got to be up there near your 120,000 to actually pull that off. And then you're looking at a good Orbitrap for that.
Thanks a lot. And the next question is coming from the US. Do you have a problem with deamination using the Smart Digest protocol at 70 degrees?
Now that is a good question because I forgot to mention it in the talk. Now, what we're looking at deamidation, yes, the temperature's up high. And yes, deamidation will occur more rapidly if it's up at a higher temperature. But what we've done here, we're not running it at pH 8. Now, deamidation occurs at high pH. So the buffer we're using, the buffer you get in the kit's a low pH digestion buffer. And that buffer there, it's about pH 7 when it's in the bottle, but once you take the temperature up to 70 degrees C, that drops to nearer to 6. So you're really doing the digest at high temperature at a fairly low pH, and because you're at low pH, the deamidation reaction's not occurring. We get less process-induced deamidation than you would do in a normal in-solution digest. The deamidation levels are very, very low, and it's the same for oxidation. We see very little oxidation due to the digest itself. So I thank you for that question because it's one that it would come up to mind. High temperature, more deamidation, but we've reduced the pH so you don't get that.
Okay, thanks. And now one additional question also from the US. Do the monodisperse columns have good reproducibilities from column to column?
Yeah, I mean, mainly it's because of the packing. It's much easier to pack something that's monodisperse. They're all going to drop down. They're going to settle at the same time. The path through them is going to be the same. So the way they pack them, it's not just a matter of packing a column easily. They go to a large, a huge optimization process to make sure the packing's good. That means it's packed at a high enough pressure and flow rate that it's not going to repack once you get it into the system. So column to column, you'll get the same amount of resin in each of the columns because it's monodisperse. Polydisperse, if you've got slightly more bigger particles, or that you get the packing, it packs slightly differently, you can get a different capacity. And capacity means a lot when it comes to chromatography. Small molecules doesn't matter so much, but when you're looking at proteins, oligonucleotides are the same. They tend to elute in a much narrower organic solvent window. And for that one, you need to take very careful control of the actual capacities of the column. These are measured under the normal standard conditions and the suitability tests, but in general, the monodispersed columns are far more reliable to pack, so they're more reproducible from column to column as well as batch to batch.
Okay. One additional question. I guess it's I could not really realize the name from which country he is coming. So, do you see method in induced oxidation when digesting at the lower pH?
No, the oxidation tends to be okay. I mean, you're in an environment where you've got a lot of TSEP there as well, and it just keeps oxidation down. We don't tend to see any added oxidation, and we don't tend to see the deamidation effects as well. So but I'd say that's a good benefit. The fact that we're not getting miscleavages is another big benefit. But I'd say the one that you've really got to look to is that it takes a very good, highly trained analyst to do an in-solution digest, and you're doing lots and lots of steps. When you've only got one step, and you don't even have to be that good at pipetting because that trypsin's in excess. From analyst to analyst, from lab to lab, it's a very, very reproducible digest. And if you're doing new peak detection in particular, you'll get more new peaks popping up from a digest because you've done the digest wrong than you've actually had new peaks. So you want really good reproducibility for that digest. If the digest is reproducible, then your assay and your multi-attribute method's much more reproducible. As soon as your digest's irreproducible, you might as well forget about doing the multi-attribute method. So although all these things are good, the column's good, the mass spec's good, your method's good, unless you're putting a good sample down that LCMS system, the rest of it doesn't matter. So the digest is very, very important. I'd say that's the best and the easiest, more reproducible digest you can do at the minute.
Okay, thanks a lot. So, looking at the time, I guess we need to getting to the end of the session. So, at the moment, there are no more open questions. So, some additional information from my side. The mentioned brochure inside the presentation are available to download direct, so immediately they are added to the webinar presentation. In some minutes, you will get a survey. So I would like to thank you in advance to fill it out. It's important for us to know your feedback for improving our webinars. Tomorrow, you will receive the recording, which you could also share with your colleagues. You will receive next week a follow-up mail with some additional information like brochures and application notes. And I would like to say thank you again to our presenter, Dr. Ken Cook. It was a really interesting presentation. And if you have any further questions, please don't hesitate to contact us at webinar@avantoscience.com. And we would like to thank you all for your attention and wish you a good day. Hope to see you soon in one of our next Avanto webinars. So thanks a lot.
All right.
Bye.